Good morning, everyone. How's it going?
Good morning.
Morning.
So to start it off today, if you guys have any specific questions about Recitation 8 or I guess anything about the class in general, you can ask them now, or we can just go over Recitation 8 as a whole. Cool. Okay, so I'll start going over the questions here. And then as I go, just let me know if you have any, or if you want me to stop at any specific one, or if you want me to go over any of the slides as well, but I'll go over the questions first. So the first one, just true or false, the forward primer anneals to the coding strand, the reverse primer anneals to the template strand. So what you have to remember is that, right, the forward primer runs 5' to 3', and so does the coding strand. And the primer has to be complementary to the DNA strand that it binds to. So therefore, the forward strand will actually bind to the template strand. So then going down to question two, match the correct order of the PCR or the steps of a PCR cycle. You'll see you just have to click one, two, or three for elongation, denaturing, and annealing, which there should be a PowerPoint slide that kind of describes that as well. Right. Yeah. You're definitely on the right track for the first two, Lakshmi. And then question three, what type of bond allows for DNA nucleotides to be built into a chain? You'll just select out of uh, actually, yeah, that second one is right, Lakshmi. Yeah. So then, right, when you're looking at the type of bond that allows DNA nucleotides to be built into a chain, right, you're on the right track there again, Lakshmi. You'll be looking at these like phosphodiester bonds. And then question four is going to be a different type of bond, which is between the two complementary nitrogenous bases. So this is just kind of all review of DNA structure. And yeah, exactly. So then understanding the structure enables us to better understand molecular cloning techniques, which are important for a bunch of different types of research. So then looking at question five, which of the following primers would have the highest melting temperature? Um, you have to remember that the number of GC bonds increases the melting temperature just because they have like that extra hydrogen bond. Yeah, GC interactions have three hydrogen bonds, whereas AT interactions only have two. So the primer that has more GC content is what it's called. Yeah. The ones that have more GC content will have a higher melting temperature. So then moving on to question six, how many copies of double-stranded DNA molecules will be produced after 13 cycles of PCR? You'll be using this 2 to the n equation. So here you'll just calculate 2 to the 13th. And this is just to show you that even after 13 cycles, where, realistically, each cycle should take like, depending on how big the DNA size, but like on average, probably like 5 minutes, you can get a lot of um, DNA molecules. Correct. Yeah, Lakshmi, you're still on the right track for all of these. Um, So yeah, looking at question six, yeah, you're calculating the number of DNA molecules you'll have. And this is helpful too, because for like a lot of different cloning techniques, you need like a good amount of DNA in order to run the experiment. So PCR is a great method for using that. Then question seven, which of the following is an appropriate component for the elongation stage of the PCR? So the elongation stage is important here for this question. Um, Obviously, you're going to need, right, like the actual enzyme to add the different deoxynucleotide triphosphate molecules. And those deoxynucleotide triphosphates are just another word for like nucleotide bases that include like the deoxyribose sugar and the phosphate group as well. Whereas in this case, there's three phosphate groups. That's where the triphosphate comes from. If you're interested, you can just look this up on Google, like why you use this specifically. But when you have those three phosphate groups really close to each other, each phosphate group has is pretty negatively charged. So there's a lot of like free energy sitting there that allows for the DNA to be built. That makes sense.
Should we consider the temperature or is it too high for that third stage?
Right. So the elongation stage is specific for a polymerase. So the polymerase itself, this Taq polymerase, it uses 68 degrees Celsius. So the 95 degrees is more for denaturing.
Okay, thank you.
Yeah, perfect. Then obviously reverse transcriptase is just another enzyme used for a different molecular cloning technique. So then looking at question 8, another true or false question. Longer bands of DNA contain more negative charges, so they move further through the agarose gel during electrophoresis. Right, so based on my video for this recitation, you'll see that this is in fact not true. Um, since the same electrical current is running through the gel when you actually like set up your PCR, you're gonna have like the same migration of DNA molecules no matter the charge, but it's more based off of size, right? So then that kind of leads into this question. So you're going to use this image. Um, and then I just kind of created like a little scenario. A researcher, you can imagine you're the researcher. You find a box of old PCR samples of unknown sizes. So what you can do with these PCR samples is you run the sample as pretty much, in like this water or buffer solution. You can freeze it at minus 20 degrees Celsius for pretty much eternity. Um, so you're hoping to find a long lost amplicon, or in other words, a PCR product to use for your new cloning project. To visualize the size of each sample, you perform gel electrophoresis and run all the samples, A through H, against a DNA ladder, which you can see here. So the DNA ladder is really important because if you didn't have this ladder here, you would just see a bunch of bands on the um, gel itself. And you wouldn't really know the sizes of it. You would just know that certain DNA fragments are bigger than other ones. So, right, you would be able to um, show here. So, all right, I see. So, Lakshmi, you're on the right page here. Liana, though, yours are flipped. And that's because the largest samples will travel the furthest, or the smallest samples will travel the furthest down the gel. So if you're looking at, lane G here, you'll see that this band is the furthest down the gel. Here's like the top, here's the bottom. And that's because the smallest DNA fragment will be able to move through like this agarose gel easier. And like this agarose gel, I like to think of it as like an obstacle course. Like you're almost running through this, I'm imagining like a trampoline obstacle course with like a bunch of these like pillars in the way, and you have to like zigzag through it. Smaller DNA fragments are going to have an easier time than larger ones. So that's why they run further. Yes. So yeah, Liana, your answers will just be flipped for this question. So if you didn't have this ladder here, you'll be able to answer this question, question 9. But in the following question, um, I like go back to the idea that this researcher or you are testing the unknown PCR products to identify the old sample, and you expect it to be around 2 kilobases. So what you're seeing here exactly, yeah, Liana, you're on the right page there. Is that there are a couple samples that are around 2 kilobases, right? So that'd be like A and F, but ultimately, out of the options that are given to you, you should see that or the answer should be distinguished. So what you're doing is just looking at all these samples, A through H, and then kind of comparing it to the ladder, which has the known sizes. So you'll see like a band that's or a DNA sample that's 5 kilobases will be like more like sample C, because it runs at about that length. And then if you're looking at 2 kilobases, you'll kind of look across and see what samples are lining up about there. So perfect. Then question 11, you're interested in amplifying a gene that's approximately, yeah, 5,000 base pairs long. How many or how long should the elongation step be? Correct, Lakshmi. Yeah. You'll know that the polymerase, pretty much every polymerase adds about 1 kilobase per minute. So per 1,000 base pairs, you'll have 1 minute of elongation time. So then here's like a troubleshooting question. A lot of the times, sorry, there's a loud sound going on. I'm not sure what that is. But sometimes you have like a lot of problems with PCR. So just imagine that um, you accidentally set the elongation step of your PCR to be shorter than it should be. You'll find that you have problems with your PCR, and then you'll just select the best answer here as the reasoning as to what would happen if the elongation step runs shorter than the appropriate time. So then question 13, this is kind of a cool application question because you can do like maternity and paternity tests using PCR because you can amplify different genes that are distinguishable between um, different mothers and fathers and the child. So a lab is doing gel electrophoresis to determine the father of this child based on the image, who is the paternal parent of the child. So what you're going to want to do, yeah, yeah, once again, Lakshmi, you're on the right track here, is you're going to kind of compare the bands with the child with the bands of the two males, and you'll find that one of the two have more matching bands than the other, indicating that male one or male two share genetic um, or some of the genes with the child. So then question 14, picture below is an enzyme that plays a vital role in performing PCR. I just wanted to show you what protein might look like when you're looking at it online. This is just like a illustration that a lot of researchers use. And this is just to help you familiarize yourself. Yeah, that this is going to be the enzyme that's used during PCR, which, yeah, Lakshmi, you're on the right track there. Um, Obviously, if you're looking at some of the other answers, specifically like PCR buffer, that doesn't have any enzymes itself. It's just a bunch of molecules. And so you want to be like kind of familiarize yourself with what a protein might look like compared to a buffer and other small molecules. So then question 15, sorry, what's your question, Lakshmi? Yes.
Uh,
Oh, if buffers are liquid, yes, they are liquid.
Okay, so but do we have to distinguish between different pictures when we are given pictures of these? I don't know how to identify a buffer or a triphosphate, I mean DNTP, if they are given to us.
Gotcha. So no, I don't think any exam questions are along those lines. Dr. Holacek just kind of wanted me to add one of these just so you can familiarize yourself with what like an enzyme might look like.
Awesome, thank you.
Yeah, exactly. But if you were looking at like you wouldn't have a picture like this for a buffer. It would literally just be like salt ions in water molecules.
Okay, awesome.
Yeah, yeah, it would look very different. And then DNTPs, they have specific structures, but they just look like nucleotides, right?
Thank you.
Yeah, of course. So then moving on to question 15, which of the following statements about PCR is correct? You'll find that a few of these are possible answers. And it just kind of shows like all the different ways that PCR can be used um, in different labs or different lab settings. So then down here, questions 16 and 17, this is, or questions 16 through 18, this is a bit harder and more, application based. A lot of times when like a researcher is developing a PCR, they have to like create these primers. And I'm realizing now that these bases down here, like this AA, that's supposed to go to this first row. So the image itself is a little weird. Um, But so the sequence for the coding strand of a mystery gene is shown below, and you know that because it's going 5 prime to 3 prime. Nucleotides are spaced out every 10 bases for legibility. Roman numerals correspond to the highlighted sequence directly below it. So here are the sequences that like you or this researcher designs, this forward primer sequence and this reverse primer sequence. So we're just asking you to kind of map the primer onto the coding sequence and like what you would expect, where you would expect the primer to come from. So yeah, Lakshmi, yeah, you're on the right track for both 16 and 17 because 16 asks which of the following sequences in the coding strand matches is the sequence of the forward primer. So in the hint I give you, the forward primers also run 5 prime to 3 prime, which is going to be the same as the coding strand. So you're just going to look for a matching sequence there. Then question 17, which of the highlighted sequence does the coding strand matches, or in the coding strand matches is the location of the reverse primer. Um, this is pretty different from the forward primer because the reverse primer Even though it's given to you 5 prime to 3 prime, on this coding strand, it's technically, um, I see. Yeah, yeah, Jeanette, this is always a tough question. I just reuse it because it's tough. Um,
Yeah, I think I was trying to explain to somebody yesterday how to figure out whether the reverse primer would go like forward. You know what I mean? Like
okay.
when you're trying to match up the sequence to one of those, whether
Yes,
you're matching it directly or matching it complementary.
I see. So for the forward primer, you'll be matching it directly, right? So this forward primer sequence, starting ACT, GAA, you'll see that one of these highlighted sequences go like use that sequence, right? So then, but with the reverse primer, yeah, it's the reverse complement is what it's called. So looking at this coding strand sequence, you're going to have to match these beginning sequences, so TAG, to the ends of one of these highlighted sequences, but it's also the reverse complement. So the reverse complement or the complement of this will be a T. Oh, that was bad. C. So you'll see, yeah, right to left. So you'll see that this sequence starts with ATC, and there was another one that did as well. Yeah, you have this ATC. So ultimately, you'll be matching it to either 3 or 4 here. Yeah.
I mean, it, sorry, it makes sense to me. I was having trouble kind of explaining it because I just finished
Yeah.
biochem, and so I'm like, well, no,
Yeah.
it just matches. It just does. And so I was having a little
Well,
trouble.
yeah, it's hard. I know, because that's how I would explain it, but not like a lot of people, these people have also asked me questions about this. That's like a hard thing also to describe because the reverse complement is like a tough thing to figure out, I think.
Yeah.
Yeah.
Thank you.
Yeah. Yeah. Thank you, Jeanette. Thanks for bringing that up. Yeah, because like in your brain, you have to like almost flip like the reverse primer sequence, like 180, and then find a complement to that. So ultimately, like I said, you'll be finding the complement of this reverse primer sequence and then going like right to left, matching it to one of these highlighted sequences. Okay, so then that'll be 17. And then in 18, you're going to find out how many base pairs the expected PCR product is. Or about. I gave like a numerical answer here, but then I gave like some leeway as well, just because it's kind of oddly spaced in this
And
question.
there is I mean, I just have need a clarification on 18. So do we
Mhm.
have to count from the primer and leave the rest on the left of the primer, the from first Roman letter, right? So
Mhm.
the A is highlighted for the primer one, right? So do
Right.
we count from there um, until the end of the primer four?
Right. Yes. So yeah, when I gave this hint that each line or the note that the PCR product contains the primers, yes, that's exactly what you're going to be doing. So for the answers that you have, which you're on the right track for, you'll be starting counting here at the beginning of primer one, and then like you said, all the way to the end of primer four.
Okay.
Right. So yeah, yeah, Lakshmi, that's the answer that I have on there for the
Okay.
key. But yeah, it's a little tough. Yes, Mackenzie. Yes. You're on the right track there. So you'll be looking like it would be, pretty tedious to actually count all these nucleotide bases one by one, but just know that each of these little segments here, even all of these like A's, even though it runs on to the next line, this is all 10 nucleotides. And then each line itself is going to be 60 nucleotides. So hopefully that makes the math easier.
Okay. Okay. Uh, yeah, your presentation for this the prep was very helpful. Thank you.
Okay, good. No, I'm glad that that helped. I just do a lot of PCRs like pretty much every week, so this is
So,
kind of a fun thing.
are you into forensic science?
No, so I'm more on the like molecular biology side. So I do a lot of molecular cloning, which I think you'll learn more about in recitation 10, but
Uh,
that has to do with pretty much doing a PCR from like an organism's genome. So you collect DNA. So it's pretty similar to forensics. You PCR
Uh-huh.
genes out of the genome, but then I put those into like DNA plasmids, um, and then uh, use those to pretty much understand like protein behavior and things like that.
Okay, thank you.
Yeah. So Mackenzie, your answer's a little bit off. Liana, yours is a tad off, but it'll be counted as correct.
So just to clarify a little bit, so when we're looking at the sequence that's given, right?
Hmm.
Are we counting each segments to see how many letters there are, or do we just know that each segment is 10?
Each segment is 10.
Okay.
Like
So
these
no
segments.
need to count those segments by any means.
Yes, correct. So yeah, ultimately, Mackenzie, I'll draw this out too. But when you're starting from up here in primer one, you'll have like this is technically 9 nucleotides just because this 10th one isn't included. So you'll have 9 and then 1, 2, 3 segments. You're gonna have 39 for that top row. That's gonna be part of your PCR fragment. Plus this whole row, which is gonna be 60.
Hmm.
Then plus another whole row, which is another 60, which is this row that includes prime or this highlighted sequence number 2. Then this whole row, which includes sequence number 3. So that's going to be another 60. Then finally, your PCR product will go all the way, oh geez, until here. So that includes 1, 2, 3, 4 full fragments, and then 1, 2, 3, 4, 5 nucleotides. So it should just be plus 45. So that's going to be your total PCR product or amplicon. So I'll leave that up for a little bit just so you guys could do the math on that. But that'll give you the correct answer. On the quiz
Okay,
itself, I
that's
gave
much
plus.
more clearer. Thank you.
Okay, awesome. Yeah. No, I'm glad that that helps. Because ultimately, these primers are just outlining the fragment of DNA that the PCR is gonna be done on. So that's why you only count between this nucleotide, this A, and then this final A here. And then if you'd like to look at that again, I'll upload this recording to Canvas under Module 5, I think we're now on. It's crazy. Awesome. So that's how you'll get question 8. And then finally, question 9, I'm introducing you to this tool called NCBI Blast. Um, so this is kind of a cool database that allows you to look up DNA sequences and find what gene it is and to what organism the PCR goes to, or
Um,
the actual sequence goes to.
could you kind of make sure that that works? I guess it wasn't working a couple of days ago.
Okay, yes,
I used it now and it works.
you
Good,
did.
perfect. Thank you.
Okay, good. Yeah, Blast. Maybe I should put this like a note on this for the future too. Blast can be hit or miss sometimes. Like, I swear, for a couple hours every day, it does not work.
Well, yeah, I think it was down for maintenance for like an entire day or something.
Oh, geez,
So,
that's super
yeah,
unlucky.
it's good that
Um,
it's back up.
yeah, just so this goes on the record, if that ever happens and you're like doing it day of, right? And Blast is down, we'll just, if the students reach out, we'll just give you guys the points because that's ultimately just an error, a technical error that you guys don't have control over. So, um, but okay, Lakshmi, I'm glad that it works. But what you'll do, there's also a link on the previous page for recitation 8 that gives you directions or like a little guide on how to use Blast. Um, you'll be doing like a nucleotide blast, which is a selection that you can make on that web page. And then you'll copy and paste this DNA sequence. This is already technically uh, in the correct format to be used for a blast query. So then you'll just blast it and then it'll come up with, thousands of these matches. But you're just going to be looking at the top one. And finding which organism it comes from. Okay, Lakshmi, to answer your question, yeah, you're on the right track for question 10 or 11, I think. Wait, 12? Wait,
Oh, yes,
yeah,
12.
12. Yeah, I think you meant 12. Um, right, because if your elongation step is shorter than it should be, the polymerase or Taq polymerase, whatever you're using, won't have enough time to actually add all the DNA nucleotides it needs to, so you're going to have a shorter amplicon than you would expect. And then that leads to an incorrect PCR fragment. And then also, the point of PCR is to go from one primer space to another and create this double-stranded DNA fragment. It's going to be pretty unstable because the ends won't be double-stranded. It'll only be single-stranded because Taq couldn't elongate both ways. And so you probably there's also a good chance that you won't have any PCR product, which is pretty frustrating after waiting around for hours for it to finish.
So are there two answers for that, or is this correct, whatever I selected?
Oh, that's correct. There's only one answer
Okay, thank you.
for question 12. Yeah. And then which of the following statements about PCR? Right. So the first one you said is correct. Yes, the second one you said is correct. And then there's also a third one, a third correct answer here. And all
Okay.
I'll say is that this answer here that says primers can only anneal to the reverse complementary site of the target DNA sequence. Primers sometimes have non-specific binding, which is also a big headache when it comes to PCR because, if you use like a 14 nucleotide sequence for a primer, there's a good chance that somewhere else in a genome, there is going to be a pretty similar sequence, and primers don't always bind 100%. Like you don't need 100% complementarity. So sometimes they'll have off-target binding.
Oh, okay. Okay. Thank you.
Yeah. But this question just also shows that one cool application for PCR is that it can be used to diagnose infections. For example, PCR was used a lot on like COVID tests, those rapid COVID tests. You're able to take like one's saliva, right? Isolate RNA molecules from it, which, the COVID-19 virus will have a specific like blueprint or fingerprint when it comes to no, footprint. That's where I was thinking of. It'll have like a specific footprint on proteins it's creating. So of course, in order to go from DNA to protein, you have RNA. So then you can actually do like a reverse transcription reaction and go from RNA to like DNA and then do PCR on that to see if you had the virus. So that's just one of the cool like applications of PCR itself.
Thank you so much.
Yeah, of course. Cool. So there were a lot of questions on this quiz. I think over like the last few sessions, we like slowly added more and more just because um understanding PCR as a whole is kind of cool. So I hope it was worth it.
I have some questions from concept quizzes. Some of them are really tricky.
Okay. I'll see if I can help with those. So it'll be module 5, I'm guessing.
Yes, please.
Okay. Awesome. So which of these um
I have like a spread out, some questions in each. So I'll
okay.
just, so
We'll go over these. Before we start going over these, Ashley, Liana, or Mackenzie, do you guys have any more questions on recitation 8? If not, we'll go over some of these concept quizzes.
Hi.
Hello.
For that last question
Yeah.
with the do we just copy and paste the entire sequence into that website?
Yes, correct.
Okay.
Yeah,
Okay.
so the link here, let me know if you could still see my screen.
Yes.
Okay, so you're gonna press on this nucleotide blast, right?
Okay.
So here you're just gonna copy and paste
Okay.
onto this area. You don't have to change any of these settings.
Okay.
I don't know when you would change these settings. So then you're gonna press blast. So after you press blast, sometimes it takes like 5 minutes, because it just adds it to this like waiting list to use this database, and it's used pretty much all around the United States. So sometimes if there's a lot of queries all at once, it takes a while. But then it'll give you like a results page that starting from the top, like your sequence was matching a specific sequence from a specific organism. For the question, you'll just say which organism it comes from. Does that make sense?
Yes, thank you.
Yeah, of course. Do you guys have any other questions? Cool. So yeah, I just think it's cool to kind of look at those bioinformatics tools. It's definitely one of the more prevalent ones that almost every lab uses. Okay, so looking at the first concept quiz, let's see. The DNA replication quiz. Okay, gotcha. So Lakshmi, do you have any questions about this
The,
quiz?
this quiz, it's kind of tricky with those numbers. We had to find all of that from the, so the first question itself, how do we know? Like not all the details were given. So that was a little bit tricky, the first question. So the 3 times 10 to the 9, right? Base pair.
Oh,
So how do we
okay.
figure it out?
Were you given the length of one base pair?
That's what I had to really deeply go through the slides and then find out, but it was not that accurate in this. So, I mean, I was like, I don't know, it was very difficult to find the information.
Okay.
So,
Yeah, I mean, how long would the genomic DNA in a single diploid cell be in your body? Okay, so you're supposed to what, multiply this by 2?
I too,
Okay.
but yeah, but still, you have some other quantity that you have to multiply. It's 3.4 something that we have to multiply. So that's how you get 2.
Okay.
Something is off there. So it was slightly difficult, that one and the last question on that particular concept quiz. It
I
was
see.
difficult.
So for this first question, yeah, I would just multiply this by 2. And then do you have the
Yeah,
length of one base pair handy? Say if,
yes, I did write it down 1 second.
no, you're good.
So it's one base pair is 3.4 A, which is 0.34 nanometers.
Okay, 0.34 nanometers.
Yeah.
I see. So,
So, but that was not given. So I had to like actually find out from the internet, like what is one base pair equal to? So will it be
gotcha.
given to us, or do we have to memorize this, conversion?
I'm not sure if you have to memorize that conversion. Jeanette, correct me if I'm wrong, but I don't know
No,
if I've ever
no,
seen, yeah.
yeah, I don't remember ever. Sometimes the concept quizzes are a little different. Dr.
Yeah.
Holchek, I don't think writes those, so they're written a little oddly. But yeah, no, I wouldn't worry about trying to memorize that number.
Okay. Okay. Thank you.
Yeah. Yeah, but ultimately you're just gonna be looking at what? So a nanometer is 10 to the minus ninth. So since this is 10 to the ninth,
Okay.
yeah, you'll find that it comes out to one of these values, like to the 10th values.
Okay. Okay. So because question 3 is a continuation of this question, after we do that 2 meters, then we have to multiply that by 50. So that's how we get that 100 billion kilometers. So one is related to the other. So it's like kind of a hit or a miss there.
Geez, okay.
Yeah.
Yeah, okay. I see
So
what you're
I'm
saying.
good with this quiz. Anybody else before we move on?
Yeah, I wonder who actually comes up with these quizzes. I thought Dr. Holichek came up with these, but thanks for clarifying that, Jeanette.
Yeah, I think that they were written back when Cogbooks was written.
Okay,
They're the exact same quizzes every time, so it's got to
I
be
see.
like part of the Cogbooks system.
Okay, that makes sense.
And
For question 2 on this one, I know
Mhm.
it's very simple, but so I understand then G and C will have 3 hydrogen bonds, and then A and T will have 2 and all that. And I
Mhm.
know that it's supposed to start from 5 prime to 3 prime.
Mhm.
But I just don't see the difference between B and C. Like, what is the difference? Just looking at the picture, I know the numbers are different, like the 5 and 3, but like,
Correct.
how am I differentiating it? I was a little confused.
So no, yeah, that literally is the only way to differentiate them.
Oh,
Each of the carbons on this deoxyribose sugar is labeled, starting with one up here. Like the one carbon is the one that's actually connected to the nucleotide base.
okay.
So if you're going like that way, you're going to find that this is the two carbon in this bottom right corner, three, four, and then off of this is like a fifth carbon. So the labeling here is the important part.
So the fifth carbon should always be 5 prime?
Yes, yeah, because that's what five prime and three prime mean. It just means like what carbon is facing out from that
Oh,
strand.
that's right. I did
Yes.
know that. Yeah,
Okay,
okay.
perfect. Okay.
Okay. All right. Thank you.
Yeah, yeah, of course. Cool. So if there's no other questions on this one, we can go to 5.2. Okay. All right, looking at DNA replication. So I know that Ashley has a question on number 4 on this quiz. Is that right? Okay. Oh, geez. If you were a primase, where would you lay down primers to replicate the melted DNA?
yep, these are difficult questions.
This is a difficult question. I've never seen this one before. So I think this kind of goes back to what you know about DNA replication, where on the technically, correct me if I'm wrong, because I haven't actually talked about DNA replication in a long time, but on the DNA, the original DNA sequence that goes 3 prime to 5 prime, the forward primer should start on like the end of that sequence because it can like um continuously replicate this 3 prime to 5 prime strand.
Um, okay, makes sense. So that
So,
D is the answer.
sorry, what was that?
That's why D is the answer. It makes sense.
I see. I think I'm able to double check the answer to that because I guess I don't really know the answer, but I think that automatically, yeah, kind of cuts out B and C because it kind of seems like in A and D, the reverse primer at some point is going to have to it's going to have to use like those Okazaki fragments if it were replicating the entire sequence. That's always confused me a bit though. I really don't like study it that much.
Actually, it makes sense, whatever you said. Yeah, because you don't want the strings to, because they are being melted. So
Uh-huh.
we the primers to attach to the portions where you said
Right.
yes.
Right. So yeah, Lakshmi, what you said was on the right track. It looks like D is the correct
Yep.
illustration. Awesome. Okay. Yeah, that's a tough question. I'm glad that you asked about that.
And question 5, also difficult question.
Oh, geez. Okay. The two oligonucleotides are allowed to anneal. What? And the DNA polymerase and all the substrates. Okay, the four DNTPs ACTG right into the mix. What will the first nucleotide incorporated in the DNA be? Wait, if the two oligonucleotides are we missing the second one?
No, the first one is that like long line, and then the second one is probably
Okay.
a primer right below it. Yeah, this is the question that somebody was asking about yesterday. So the, this one I've been through a couple times. It's a pain.
To anneal. This just feels like an incomplete question. If the two
Yeah.
oligonucleotides are allowed to anneal, yeah, who was paid to actually write these questions? The two oligonucleotides are allowed to anneal to what? Okay. And the DNA polymerase and all substrates, okay, are added to the mix. What will the first nucleotide incorporate to the so it seems like this first one up here is supposed to be maybe the
T.
forward primer. Yeah, so here it would be T.
I think the bottom one is the primer, and the top one is some like the way that you had it, but like a much shorter DNA strand.
Yeah,
So
almost like the template, I guess.
yeah, I mean it doesn't actually say
So that would make sense, but then that would make the answer C, which is not the right answer.
no. If you find so that has got to be like a reverse primer or
Yeah,
something.
it must be because then you're looking at this, you're thinking,
Well, it's like if you match that thing up, if you match its complement up to the one above,
okay.
then the very
Oh, I
next
see.
pair that would be
Wait.
added would be, it's like down towards the end. If I remember correctly. I think I wrote it down some, oh, no, I erased it. Yeah, so it's like right in there, um, like going backwards, it's like C, C, T, G, G. Yeah, so
C,
then it
C,
goes
T.
up to the T, and then the very next one is A. So what would be added would be at the
Oh,
T.
wait.
It's a really messed up
T.
question.
Okay, C, C, T, G, G, I, C, T, G, T, G, A. So it'll be this that it actually aligns to, huh?
Yeah. So then the very next one that would be added in that direction would be a T to complement the A.
But how are we supposed to, do we have to, like, it doesn't say whether it is a prokaryote or a eukaryote, right? So we don't know whether it is a 35 consensus or 10 consensus that we have to remove or whatever it is, right? The sequence. So it's so confusing as to where we have to start to find the answer for this.
Yeah, no, I totally agree. It's a really poorly written question that makes absolutely no sense.
It is. Yeah, that's frustrating. The heck? Okay, so ultimately, you're just going to be looking at this nucleotide or this one.
Okay.
Yeah. So the nucleotide that's complementary to the A will be the first one incorporated into the DNA. Just because this will bind. Oh, where did I have it? Oh, I lost it. It was this, I think? Wait, no. Anyways, it's so since it's a reverse primer, this one will be read first, so it's whatever's complementary to the adenosine.
Oh, so we just have to find the sequence that is reverse complementary to this for primer.
Yes, which I think was TG. Wait.
Yeah, T next to it.
Yes.
Yeah.
Awesome. Okay, so it'll be this. So then when the polymerase is going 5' to 3', and it's like, let's see, it's going this way, it's going to read this nucleotide first and add it on first.
I'm sorry to ask this question. So just a thing. So when I went through these 6 or 7 videos posted by Dr. H, but
Mhm.
some of these concept questions are not being covered there. So but we are asked these questions. So it's like puzzling because we don't have the proper materials to answer these questions. Thank goodness there are 2 tries, but still it's kind of very very unrelated, like, so the
Mhm.
video has those questions and they are in depth, but these concept questions, like, they are totally, some of these questions are unrelated, like the questions that we have discussed, like the diagram, right? It was just
Right.
a diagram when Dr. H went through in the videos, but
Mhm.
it didn't have exact problems to discuss about how we have to find out what those answers or right answers or right way to answer those questions were. So
Mhm.
it's uh, It's kind of puzzling. It's actually, as you guys said, it was from the cog books. I think you had to read those chapters in the textbook, and they were assigned these questions. But since we don't have the textbook, we don't know actually where we are looking at.
I see. Yeah, I mean, um, I'm not sure how Dr. Holacek created her lecture material. If she, I mean, it sounds like she didn't use all of these questions to kind of create her slides, or else they would all be covered. But if, I mean, as you go along, if you're not sure about some of the stuff, you can definitely use Google to try to figure that out. But I don't know, Jeanette, if you have any recommendations since you've taken this class previously.
Um, I mean, I shouldn't say it, but I usually use GPT and just plug the question in and have it explain it to me so that I'm not just like getting an answer, but that I actually, I'm getting like the explanation of why, if that makes sense. Um, I know I know it's really, really frustrating, but the concept quizzes tend to go more to the like readings within, but kind of take it to a different level that isn't anymore like it's not included in the course anymore. So I think she's working on the last I heard she's working on getting rid of the cog books like stuff completely, but for right now, um if you can find the answer by either Googling it or, coming on Discord and asking one of us and we can kind of walk you through it. But yeah, it's a lot of this stuff just isn't in the lectures.
Yeah, it's not covered. Yep, yep.
Yeah,
Thank
it's frustrating. I'm so sorry.
Thank you. That's what I asked Dr. H in the first week because it was total disconnect from the concept quizzes
Yeah.
and the problems. And thank goodness recitation, you guys, I mean, TAs post a lot of information that helps. But for the concept quizzes, they are totally scattered. So it's really difficult to focus. And these 8 or 7 were due yesterday, and we just had our second exam. It was really tough.
Oh, definitely. Yeah, that's a very quick turnaround. Um, but definitely make sure you take note of that on the course evaluation because Dr. Holacek takes those very seriously.
Okay, sure, sure, definitely.
Yeah.
And I have a question on what is it? Causes of mutations and direct repair of DNA damage.
Okay.
And at least if we can go through that together, I think we'll have some more insight on that. Question number 4, again with the diagram.
Okay, I see. So it's question number 4 on this one.
Yeah.
Oh, okay. Aflatoxin. Okay. The graph shows the results of an Ames test. I'm not really sure what an Ames test is. To identify the effect of several concentrations of, okay, using a couple of his salmonella strains. Okay. While TA100 is a strain sensitive to reversion mutations by base substitutions, TA1538, okay, is sensitive to frameshift mutations. Based on the results, what kind of mutations can aflatoxin B1 lead to? Okay, so aflatoxin, can't tell if it's like a drug or something, but so what you see here is that TA100 is one of the salmonella strains used. As the amount of aflatoxin B goes up, revertant colonies, the number of revertant colonies, which I'm not too familiar with. I'm actually going to look that up. You're right. These questions are kind of crazy. Colonies that have regained a lost function due to a secondary mutation. There are colonies of bacteria that have mutated back to a state where they can synthesize a required nutrient. Okay. As you see, like when I look things up, I know like Jeanette said, we're not supposed to use AI to, of course, write anything, but it's a very helpful tool for actually summarizing material. And as you can see here, it gives you like
Right. Okay.
helpful links to go to. So
Yeah, the answer choice is all except frame shift mutations, but it's kind of a tricky question from the diagram that we can. It's not like a normal graph, like we can just deduce anything. It's kind of
Right. So it seems like, so since TA 100, it looks like gains its function back. The number of revertant colonies increases, right? So since it gains its function back, we can assume that this aflatoxin B1 leads to whatever mutation
uh-huh.
that gives TA 100 its function back. So it says while TA 100 is a strain sensitive to reversion mutations, I would assume that this mutation is what aflatoxin B1
Oh,
gives.
uh-huh. Okay. So yeah.
So you said these were due last night?
Yes, they were due last night.
Do you have the answer? Like, were you given feedback already?
No, actually, yes, yes. That was the answer. All except frame shift mutations.
Oh, cool. Okay.
Yeah, because we had two tries, so that was a good one. But still, these these are these are really tough.
They are really tough. Honestly, it feels like I'm back in high school taking standardized testing and I'm just given like random questions.
Yeah,
So,
and yeah, because those, I mean, that's all. So we don't know where to concentrate because already we have a lot of stuff to study, and these concept quizzes are giving us different kind of other materials that we have to focus on. So I don't know, it's kind of a scary thing. So now we have to focus on all of these things together, but we are unclear because everything has to be discussed, and then we prepare ourselves for exam 3. So we don't know where we are because there are too many um, you know I don't know, unclear stuff here.
definitely. Based on the first two exams, did the concept quizzes help much in
um,
preparing for the types of questions you saw?
Only the ones, I mean, I think the ones that were little bit closer to what we had on the exam was what Dr. Holchek discussed with us in the office hours.
Okay.
Uh, those were close, but not obviously not everything was relevant. But yeah,
Hmm.
yeah,
Okay. Yeah, I mean, if you've found that only the questions that Dr. Holacek goes over are, important for um are applicable to the exam questions, then that's probably going to be the same thing for exam 3 and like the final.
okay, okay,
Um, yeah, I mean, as I think you're hitting it right on the dot where you're saying that these concept quiz questions are, a bit further away from what the activities, recitations, and exam questions might be going over.
right,
So I would just use the concept quizzes on just kind of expanding your general knowledge, but
okay.
might not be as applicable to things that are important for your grade. And I
Okay.
forgot that you had two attempts on every quiz. So if you're given the correct answer after the first attempt, hopefully
Yeah,
that
yeah,
that
that's
helps.
only for concept quiz. For recitation, we don't have second attempt. So, but for concept quizzes, yes, we have two attempts.
Okay, that
Uh,
sounds good.
mm,
Yeah, and then for the recitations, of course, us TAs make them, so that's different also than what Dr. Holacek probably goes over. But she
uh-huh.
does make sure that we have specific questions on the recitation quiz that
Uh-huh.
are a bit more applicable.
Right, right.
So,
And your videos help, so that really helps us a lot for recitation. And it is more
okay,
number of points than concept quizzes. Yeah.
okay,
And so I thank you so much, Luke. And I emailed you before our office hours. I emailed you my activity 6
okay,
and recitation 8. So you can ignore recitation 8, and if you can just look at activity 6, that will be helpful for the feedback.
okay. That sounds good. Yeah, I'll let you know if there's anything glaring on what you have so far.
Thank you. Thank you so much.
Yeah, yeah, of course.
Thank you.
Yeah, bye. Thank you. Ashley, how's everything going? Do you have any more questions on the any of this material? All good? Okay, cool. Of course, if you have any questions that come up after this, feel free to reach out. Yeah, definitely. Google is going to be your best friend on these concept quizzes, I guess. Cool. So if you don't have any further questions, I hope you have a nice rest of your week, or at least rest of your Monday. However much of it you have, depending on where you live. Yeah, of course.